Hello class, today I will talk about tumor suppressor gene. Why is it important to talk about tumor suppressor gene? Because it inhibits growth, division of cells, and prevent formation of tumor. In order to have successful mechanisms of tumor suppressor genes, we will need retinoblastoma, which is an eye disease that happens at early age, and transcriptor growth factor with E2F and P53 genes. In P53 genes, we will need P21 and CDK cycling. Tumor suppression example is like a car brake stop car moving on. The brake car cannot stop. Now I will go more in depth about the mechanisms of tumor suppressor genes. The cell reach to G1 and move on to S phase, which is DNA synthesis, need to build a protein to carry out DNA replications. So then transcription growth factor with E2F will bind to sequence of DNA, which transcription and translation will occur to have protein. At G1, E2F stop build protein for DNA replications. In cell, E2F bind to RB inactive E2F and stop cell DNA from replications, which means G1 we will stop at G1 and not move to S phase. RB phosphorylate, which is mean add the phosphate group, and E2 will bind to a sequence of DNA as usual, and then transcription and translation will occur. But in this case, G1 will move to S phase. RB mutated cannot bind to E2F whether we have the phosphate group or not. It will not function anyways. But the E2F still allows cells to move to S phase. As we talk about the tumor suppressor gene, it would not complete without P53 gene. P53 genes is stop tumor from developing. 50% cancer have mutation in P53 gene. Besides that, P53 genes also activate P21 and regulate its own expression and bind to broken DNA. Now I will talk about the mechanism of P53 gene. It will either cell cycle address, which is hold the cell cycle at G1, will not move to S phase, or apoptosis, which means it kills itself. Another thing P53 gene can do is can produce a protein turn on 21 to attack CDK and cycling, which will lead into no cell division, which is no function. So the cell remain in G1, will not move to S phase and go to apoptosis.